Hi, everyone. So we are moving on from chapter 12, where we talked about mitosis, and now we're moving on to chapter 13, where we're going to talk about meiosis. So chapter 12 is mitosis, and chapter 13 is, of course, meiosis. And we're going to talk about the differences between these two. As I've said in my previous videos, if you're watching this in the fall of 2020, you do have a quiz on Friday. And that is October 9th, if this is the fall of 2020. And that quiz is going to cover chapters 12 and 13. All right. So where are we headed in this chapter? Well, first off, I want to talk about what meiosis is. And we're going to talk a little bit about why we need meiosis. Why does this process even exist, okay? And we're going to compare it with what we already know about mitosis, okay? And so we'll work on making sure you don't confuse these two processes. Then we're going to talk some about how meiosis promotes genetic variation. We'll also highlight why genetic variation is important in a population, right? And then we'll talk a little bit about what happens when things go wrong. So there are some diseases that result from meiosis going awry. One of the most notable that you would have heard of is something called Down syndrome. And we'll talk about how that happens as a result of mistakes in meiosis, all right? So in thinking about mitosis versus meiosis, I want us to think about why we even need two different processes. So let's back up for a second and remember what it is that mitosis with a T, what does mitosis accomplish? Well, mitosis makes two exact copies of a cell, okay? Mitosis with a T makes two exact copies. So what do we mean by that? Well, essentially what that means is that the number of chromosomes that exist in the daughter cells that are there after division is the same as in the original cell. So if you started out with 4 chromosomes, after mitosis, both daughter cells would have 4 chromosomes. Are you with me? So these are exact copies of the original cell. And we find this in what we call somatic cell division. Now, what do we mean by somatic cells? Somatic cells are what I think of as body cells, okay? Skin cells, liver cells, brain cells, okay? Now, let's think about gametes. Gametes are what we call sex cells. So if we're talking about humans, what we mean by that are egg and sperm cells, right? And if we wanted to get really, really specific, we would talk about oocytes. Those are egg cells. And we would talk about spermatocytes. Okay, now let's think about why we need mitosis versus meiosis. Meiosis generates sex cells. Mitosis with a T generates body cells. So let's think about this for a second. Let's think about humans and how many chromosomes you have in your cells. So inside your cells, let's see here. This is a picture of the chromosomes that exist inside your cells, okay? This picture has a name. A picture of chromosomes is called a karyotype. And the way you generate this karyotype is some scientist looks at dividing cells through a microscope, takes a picture, and then you cut that picture up and you match up all the chromosomes. And what I hope you see here is that you have 23 different kinds of chromosomes, 1, 2, 3, et cetera, et cetera. But each of these different kinds of chromosomes has 2 copies. So you have 2 copies of chromosome 1, 2 copies of chromosome 2, etc., etc. So overall, you have 46 chromosomes in your cells, okay? And that, again, is the result of 23 different types of chromosomes, each of which has 2 copies. Now, I want you to envision that, let's pretend that your egg cells had 46 chromosomes, and a sperm cell, oops, let me erase this, and a sperm cell had 46 chromosomes. Oops, come on, there we go. Now let's just say a fertilization event occurred where this egg fused with this sperm. Well, what would happen? Suddenly we would have 46 plus 46. And if you do that math, that comes out to 92 chromosomes. Well, geez Louise, what if this 92 chromosome baby mated with another 92 chromosome baby and their egg and sperm fused? Then you would have, let's see here, 184 chromosomes. And you could go out and out and out and see that if you allow sperm and egg to maintain their original number of chromosomes, then you end up with crazy mutant babies with increasing numbers of chromosomes in each subsequent generation. That's not what happens. Instead, what happens is the process of mitosis takes a 46 chromosome early egg cell and reduces those chromosomes down to 23. So then you have an egg cell that has 23 chromosomes. A similar process, happens during sperm development where 46 chromosomes are reduced down to 23, and that creates a sperm cell. Now, if we have a fertilization event between this egg and this sperm, we restore the original number of 46 chromosomes. Half your chromosomes came from mom and half your chromosomes came from dad, right? All that makes sense now, doesn't it? So the reason that we need meiosis as a process is because meiosis is a reductional division. And it decreases your chromosome amount by 1/2. So essentially, what meiosis is going to do is it's going to split up these pairs for each of these chromosome pairs, okay? And we'll talk about how that happens. Here's a little overview for you. If we were talking about mitosis, which makes 2 exact copies, you would start out with 46 chromosomes in the parent cell, and the 2 daughter cells would also have 46 chromosomes. This would happen in your somatic cells. So any of your body cells, skin cells, liver cells, stomach cells, you get where I'm going, right? But if we're talking about developing your gametes, if we're talking about egg and sperm production, you start out with 46 chromosomes. But then in meiosis, we end up going through 2 rounds of division so that you end up, instead of 2 exact copies, you get 4 copies, each with half the number of chromosomes. Okay? And we're going to walk through this process. All right, let me erase this right here. Let's think about chromosomes a little bit more before we move on to talking about meiosis. So before we talk about meiosis, it's going to be important for us to differentiate between 2 terms, haploid versus diploid, okay? A cell that is haploid has 1 copy of each chromosome, okay? So here we see a pink chromosome, a blue chromosome, and a green chromosome. And you can tell that they're all slightly different because they're slightly different sizes. This means that they have different amounts of DNA in them. And these probably code, well, no, they would certainly code for different genes. So maybe there's a gene for eye color on this one. Maybe the gene for hair is on this one. And maybe the gene for skin tone is on this one, okay? So these are 3 different chromosomes. They're gonna have 3 different sets of genes on them, all right? Haploid cells have only 1 copy of each chromosome. Diploid cells, in contrast, have 2 copies of each chromosome. So here you can see we've got 2 copies of the pink one, 2 copies of the blue, and 2 copies of the green. This is the situation in your somatic cells. This is the situation in your gametes or your sex cells, okay? So let's go back here to this slide above here. And what you can see is that this karyotype here is from a diploid cell. How do I know that? We've got 2 copies of each chromosome. Now let's look at my little overview here of mitosis versus meiosis. In mitosis, we start with a diploid cell with 46 chromosomes. That's the situation that we see up here, okay? And we end with 2 diploid cells, each with 46 chromosomes. In meiosis, we're going to start out with a diploid cell that has 2 copies of each chromosome, but we're going to end with 4 haploid cells that each only has 1 copy of each chromosome. All right? Now let's talk a little bit further about something called homologous chromosomes. All right? So these are 2 copies of this pink chromosome here. So this is called a homologous pair. Homologs, homo means like, right? So those are homologous pair. That's a homologous pair. 2 copies of the blue, these are homologs, or a homologous pair. Those are synonyms. And here we have 2 copies of the green, also homologs or a homologous pair. All right. Looking here, I also want to talk a little bit more about our anatomy of a chromosome, because we talked about this in our last chapter, but again, I don't want anyone to get confused. So here, I hope you can see that I have a homologous pair. And I know that because these chromosomes are the exact same size and shape. Now, I got this from a different image, so they've colored them different colors, but you can see that they're the same size and shape. I know that they're homologs. If I go back here, these are the same size and shape. I know that those are homologs, okay? Looking at this homologous pair, we have on each of them, we have a centromere. Remember, the centromere acts like a staple to hold 2 copies together. Ugh, I'm running out of room here. Remember that this is 1 copy of DNA. This is a 2nd copy of DNA. This is a copy of DNA, and this is a copy of DNA. So both of these chromosomes have been duplicated. We have 2 chromosomes, but 4 copies of DNA. Also, remember that each one of these individual little legs here is a chromatid. And these 2 together are sister chromatids. okay? So make sure that you understand the various parts of a chromosome, because I'm going to be referring to them as I explain how meiosis works. Now, another thing that I wanted to point out, so here we can see that we have a homologous pair, right? And we know that because they're the same size and shape. These two are sister chromatids. These two are sister chromatids. They're joined at a centromere here and here, all right? Now, I wanted to point out something to you. You can see that as you go through, this is a human karyotype. As we look at our various chromosomes, they're kind of, some are bigger and some are smaller, and we have them arranged here in order of size, with the biggest as number 1 and the smallest as number 22. And then we get to the very end and these chromosomes start to look different, okay? X chromosomes look like this, and then Y chromosomes look like that. So these are what we call the sex chromosomes, and they determine your biological sex. Okay, so you guys probably know that if you have one X chromosome and one Y chromosome, then you are male. And if you have 2 copies of the X chromosome, you are biologically female. Okay. Now, what's interesting is if you have 2 X's, then you have a homologous pair. But if you have one X and one Y, those are not homologs. They have different genes on them. The Y chromosome is essentially the set of instructions for how to make male sex characteristics, male genitalia, etc. Okay? So these sex chromosomes are different from the rest of the chromosomes. The rest of the chromosomes are what we call autosomes. And those are the same in everybody in terms of their size and shape. Okay, whether you're male or female, you have autosomes that look like this. But if you're female, your sex chromosomes are XX, and if you're male, your sex chromosomes are XY. Okay? All right, now that we understand a little bit about chromosomes, we can have a conversation about what happens during meiosis. The big deal with meiosis, which remember that meiosis is a reductional division. The big deal with it is that it separates homologs. So I'm going to give you kind of an overview. Reductional division. Sorry, my stylus is crazy tonight. Okay, so I'm going to give you kind of an overview of what happens during meiosis, all right? So I'm going to draw for you. We're going to start out with a parent cell here. I'll make that kind of big. And what we're going to do during meiosis is we're going to go through 2 rounds of cell division. This parent cell is going to divide into 2 cells. And then those cells are going to divide into 2 cells. Now let's insert some chromosomes. And what I'm going to do is I am going to make the maternal chromosome red, because that's what your book does. And I'm going to make the paternal chromosome green. Sorry, blue. I don't know why I said green. Now, I hope you can see that I intend for these to be 2 homologous chromosomes. They are the same size, they are the same shape, okay? In my little diagram, I have them joined at the center here, so it's kind of understood that there's a centromere there and a centromere there. And you can see that you've got 2 sister chromatids on the red, 2 sister chromatids on the blue, okay? So what meiosis does is it separates homologs. So in this example, the maternal chromosome gets separated in the first round of division from the paternal chromosome here. Okay, and remember that this is actually 2 copies of the same DNA, and this is 2 copies of the same DNA. And so what's going to happen next is this round of division is going to pull the staple apart. So you have one copy in each there and one copy in each here. Now let's look at what we've done. This section of meiosis is called meiosis 1. What basically, we're going to go into some more detail, but what's basically happening in meiosis 1 is we're going to have some spindle fibers that attach, and they're going to pull this homolog that way and this homolog that way. Cytokinesis will pinch in, and you'll have a situation where you have an intact copied chromosome with its centromere intact and its sister chromatids intact in your first cell here. And in this cell here, you're going to have, again, an entire copied chromosome with the sister chromatids and the centromere intact. Then this round here is called meiosis 2. And meiosis 2 looks very similar to mitosis, because if you remember in mitosis, what happened was microtubules attached here at the centromere and pulled in opposite directions, eventually causing this to split down the middle. And so you end up with one copy of DNA here and one copy of DNA here. The same is going to happen here, where those microtubules are going to pull in opposite directions, cause a centromere to split, one copy and one copy. So the way that we accomplish our reductional division in meiosis is by inserting this extra step, okay? So it's meiosis 1. This is where the reduction occurs. Meiosis 1, let me say that again. Meiosis 1 is where the reduction occurs, okay? Because what happens here is the entire homolog gets pulled apart. The entire homolog gets pulled apart. And the secret is going to be that when the chromosomes line up, remember that in mitosis with a T, mitosis. In mitosis with the T, the chromosomes during metaphase, they lined up in single file fashion, and then they got pulled apart. In meiosis with an O, during meiosis 1 metaphase, the chromosomes line up 2 by 2 with the homologs lining up next to each other, and then they pull apart. And so that's really the key to the reduction that we're going to see in meiosis 1. And then meiosis 2 is really just like mitosis, okay? So let's look at this in a little greater detail. We're going to look at the individual stages. So we're going to start with the parent cell, and that parent cell is going to be in interphase. And if you recall from our last lecture, interphase is actually several steps. It is the G1 phase, it is the S phase where the DNA is copied, and it is the G2 phase, okay? And if you'll also remember, during interphase, the DNA in the nucleus does not exist as chromosomes. It is just a loosey-goosey mass of chromatin. Review question, what's chromatin? How is it different than the DNA by itself? All right, think about that and see if you can answer that for yourself. So here is a picture of what an interphase cell looks like. You can see that we have uncondensed chromosomes, and they are replicating in the parent cell. Now, as this parent cell moves into meiosis 1, we're going to enter prophase, okay? So in prophase, kind of similar to what happens in mitosis, we're going to see the chromosomes condense. We're going to see the spindle apparatus form, and we're going to see the nuclear envelope begin to break down, okay? But something unique is about to happen in the prophase of meiosis 1. And the unique thing that's about to happen is called synapsis. Synapsis is when the homologous or the homologs, the homologous pair, homologs match to one another. Okay, so here you can see that these homologs have found one another. These homologs have found one another, as have these, okay? Again, the red is to signify a chromosome you got from mom, and the blue is to signify a chromosome you got from dad. But you have two copies of that chromosome, one from mom, one from dad, right? So these homologs have lined up. Another neat thing that happens at this phase, and I'm going to come back to it later, but I just want to point it out really quickly. I'm hoping that you can kind of make out that on some of these, the chromatid legs of one chromosome have gotten tangled up with the chromatid legs of another, okay? And so this is where something called crossing over is going to occur. Crossing over happens during prophase, and I'm going to come back to that in a later slide, okay? So in prophase, let's just review. I'm going to draw a little line here just to review the things that have happened in prophase of meiosis 1, because this is an important step. So the first things that are going to happen are kind of review for us. We know that in prophase, chromosomes are going to condense, so the chromatin is going to pack into those little X shapes. The spindle apparatus, remember this is our spindle apparatus here, it's going to begin to form. We're also going to see the nuclear envelope here break down. What is new is that synapsis is going to happen where the homologs match with one another, okay? That's going to lead us into metaphase. Now remember, in metaphase, chromosomes migrate to the middle. All right, so we're seeing our chromosomes migrate to the middle, but they're migrating in pairs. They're lining up 2 by 2 instead of single file. That's another important difference, okay? So lining up 2 by 2, here it says movement of bivalence. What they mean by bivalence are the pairs, the homologous pairs. Movement of bivalence is what happens in metaphase 1, so they should be in the middle. Anaphase 1, this is where the reduction happens, people. This is where the party is, okay? Reduction, because what we're gonna do in anaphase 1 is we're gonna separate the pairs, the homologs separate. So you can see here that what has happened is our microtubules have grabbed this centromere, they've grabbed this centromere, and they've grabbed this centromere, and they're pulling the entire thing across, okay? The entire X, if you will, is getting pulled across. That's different than mitosis. Go back and review that if you need to, okay? This is where the reduction happens because this is where we are going to separate homologs. All right, let's move on. Now we're going to get to telophase 1. In telophase 1, what's going to happen is that the homologs are going to be at opposite poles. The spindle apparatus disassembles, so the microtubules go away, and cytokinesis occurs to separate out the cell membranes. And so at the end of meiosis 1, you go from being a diploid cell to being a haploid cell, okay? So the reduction has happened in meiosis 1. But if you look here, you can see that each of these little chromosomes has 2 copies. It's got a centromere with the 2 copies stapled together, okay? There you are. So the next step, meiosis 2, is going to look really familiar. It's very much like mitosis because what we're going to do is we're going to break apart that staple and separate the copies. So let's look at what happens. Prophase 2, we're going to see our spindle apparatus reform. Oh, one quick note. Notice what did not happen here. The nucleus did not reform. And that's because we're going to move right into this second process. So in this second process, our chromosomes are already condensed and the nucleus is already gone. So all we have to do is we've got to reform our microtubules, okay? Then in metaphase 2, our microtubules are at opposite poles. They're going to attach to centromeres here, here, and here, 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 and here. They're going to start tugging in opposite directions, so that direction and that direction. And as that happens, you can see that we break those centromeres apart and we pull those copies to opposite sides of the cell. And so that happens during anaphase 2. And then in telophase 2, what's going to happen is we're going to have the nuclear envelope reform. We're going to have the chromosomes relax. We're going to have the spindle apparatus disappear. We're going to have cytokinesis occur. Okay, let's watch a little video that shows us this. Let me change my screen share. Here, okay, we'll do desktop 1. Let me share that. All right, this is a video from your text that shows you this process in an animation, okay?
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
All right. Let me go back to our original.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis.
There we go. Okay. All right, everybody. So hopefully that helps you understand meiosis as a process. So where we're going to pick up in part 2 is we're going to talk about a process called crossing over. We're going to talk about how meiosis generates genetic variation, and we'll talk about what goes wrong during meiosis. Talk to you soon. Bye-bye.